Look around you. Every city, every computer program, every machine we build relies on code. Binary code, ones and zeros. But nature engineered its own software billions of years ago. A code so dense that a single gram of it can store up to 215 petabytes of data. I'm talking about DNA. Inside nearly every single cell of your body sits a microscopic manual containing roughly 3 billion letters. This manual dictates the color of your eyes, how your heart pumps blood, and how your body fights off disease. For thousands of years, this manual was completely locked. We knew it existed, but we couldn't read it. That all changed with a breakthrough technology called DNA sequencing. Today, we aren't just looking at the cover of the Book of Life, we are reading it word for word, letter by letter. We can trace ancestry, catch genetic diseases before symptoms appear, and engineer crops to survive droughts. In this video, we are breaking down exactly how DNA sequencing works. We will travel from the pioneering experiments of the 1970s to the massive high-tech machines of today that sequence millions of genomes simultaneously. Whether you're a student, a science enthusiast, or just curious about the technology shaping the future of medicine, strap in. Let's decode DNA. Before we look at the machines that read DNA, we need to understand what they are actually reading. DNA stands for deoxyribonucleic acid. Think of it as a long, twisted ladder, the famous double helix. The rungs of this ladder are made up of 4 chemical building blocks called bases. We represent them with 4 simple letters, A, T, C, and G. A stands for adenine. T stands for thymine. C stands for cytosine. G stands for guanine. These letters follow a strict rule called complementary base pairing. A always bonds with T, and C always bonds with G. Because of this rule, if you know the sequence of one side of the ladder, you can perfectly predict the other side. Now, why does the order of these letters matter? Because the order determines everything. A sequence like AATGCC tells a cell to build one specific protein, while changing just a single letter to AATGGC could completely alter that protein's function, or stop it from working altogether. DNA sequencing is simply the process of figuring out the exact order of these letters across a specific strand of DNA. But there's a massive catch. DNA is incredibly tiny. You can't just put it under a regular magnifying glass or light microscope and read the letters. It's physically impossible. So, scientists had to get creative. Instead of trying to look at the letters directly, they figured out how to make the DNA copy itself, and copy itself in a way that betrays its own secrets. The story of reading DNA truly begins in 1977. A British biochemist named Frederick Sanger invented a method that would dominate the scientific world for 3 decades. It's called Sanger sequencing, or the chain termination method. Sanger's trick was brilliant. He realized that to read DNA, you need to watch it build. Imagine you are building a wall with regular red bricks. As long as you use red bricks, the wall keeps growing. But now, imagine someone mixes in a few special blue bricks. The moment you place a blue brick, you are blocked, you cannot place any more bricks on top of it. The wall terminates. Sanger did exactly this at a molecular level. He took the DNA sample he wanted to sequence and mixed it with the normal ingredients for DNA replication, enzymes, primers, and plenty of normal A, T, C, and G bases. But then, he added a tiny amount of modified bases called dideoxynucleotides, or chain terminators. If a normal base is a regular brick, these modified bases are the blue bricks. Each type A, T, C, or G was tagged with a distinct fluorescent dye or radioactive label. As the replication reaction occurs in the tube, the enzymes build new DNA strands. Every now and then, by pure chance, a modified stop base gets incorporated instead of a normal one. The reaction halts immediately. Because this happens randomly millions of times in the tube, you end up with a collection of DNA fragments of every single possible length. One fragment might be 10 bases long, another 11 bases long, another 12, and so on. Crucially, every single fragment ends with one of those glowing stop letters. The DNA fragments are injected into a microscopic tube filled with a gel and exposed to an electrical current. Because DNA has a negative charge, it moves toward the positive end. Smaller, lighter fragments race through the gel quickly, while longer, heavier fragments lag behind. A laser at the bottom reads the fluorescent color of each fragment as it passes by. If the smallest fragment passing the laser glows blue, representing C, the first letter is C. If the next smallest glows green, representing A, the second letter is A. By reading the colors from smallest fragment to largest fragment, the machine reconstructs the sequence perfectly. Sanger sequencing was a masterpiece of biochemistry. It was incredibly accurate, over 99.99% correct. It was the absolute backbone of the famous Human Genome Project, which mapped the first complete human genome, finishing in 2003. But it had a massive flaw: it was slow and incredibly expensive. The Human Genome Project took 13 years and cost roughly $2.7 billion. To sequence millions of people, or to use this in everyday hospitals, we needed a revolutionary upgrade. If Sanger sequencing is like reading a book letter by letter, NGS is like ripping the book into millions of tiny pieces, reading all those pieces at the exact same time on a massive computer, and then stitching them back together. The undisputed king of this generation is a technology called sequencing by synthesis, popularized by a company called Illumina. The process works in 4 clear steps. Step 1 is fragmentation. We take the genomic DNA and shatter it into millions of short fragments, usually between 100 and 300 base pairs long. Step 2 is library preparation. We attach short, synthetic pieces of DNA called adapters to the ends of these fragments. These adapters act like physical keys that allow the DNA to stick to a specialized glass slide called a flow cell. Step 3 is cluster generation. Once the DNA fragments stick to the flow cell, they are amplified using a process called bridge amplification. This creates dense microscopic clusters, where each cluster contains thousands of identical copies of one specific DNA fragment. Step 4 is the sequencing reaction itself. This is where the magic happens. The machine floods the flow cell with special fluorescent bases and DNA polymerase. Every time a base binds to a cluster, it emits a tiny, characteristic flash of light. A highly sensitive camera hovering above the flow cell records millions of these flashes simultaneously across the entire surface of the slide. As the machine builds the strands base by base, it records the color changes over time for every single cluster. Finally, powerful computer algorithms take over. This is the bioinformatics phase. The software processes the raw video data of the light flashes, converts them into millions of short text strings called reads, and maps them against a known reference genome like fitting puzzle pieces onto a picture template. The sheer scale of NGS is hard to comprehend. Instead of sequencing one single fragment at a time like Sanger, a modern NGS machine can read billions of fragments at the exact same time. Because of this massive parallel processing, the cost of sequencing a human genome plummeted from $2.7 billion down to under $200 today, and it takes less than a single day to finish. But even next-generation sequencing has an Achilles heel. It can only read short fragments. Imagine you are trying to solve a jigsaw puzzle of a clear blue sky. If all the pieces are tiny and look identical, it's almost impossible to figure out where they go. The human genome contains massive regions of highly repetitive text, thousands of letters where the sequence is just AG over and over again. When NGS chops these regions into short fragments, computers cannot figure out where they belong. To solve this, scientists developed third-generation sequencing, also known as long-read sequencing. Instead of chopping DNA into tiny bits, third-generation technologies read incredibly long single strands of DNA, tens of thousands of bases long, in real time. There are 2 primary platforms dominating this space. First, there is PacBio SMRT sequencing. This technique uses microscopic wells called zero-mode waveguides. An enzyme sits at the bottom of the well, and as it synthesizes DNA, it detects light emissions from individual molecules in real time with stunning accuracy. Second, and perhaps even more futuristic, is Oxford nanopore sequencing. This method completely throws out lasers, dyes, and chemical synthesis. Instead, it uses a tiny synthetic membrane embedded with microscopic, biologically engineered protein holes called nanopores. An electrical current is passed across the membrane. When a long strand of DNA is pulled through the nanopore, the physical bulk of each base A, T, C, or G partially blocks the opening, disrupting the electrical current in a highly specific way. As the DNA slides through like a tape measure, a sensor measures the fluctuations in the current. A computer trained with machine learning interprets those electrical ripples and translates them back into DNA letters instantly. Because nanopore technology doesn't require complex lasers or cameras, the sequencing hardware can be incredibly small. Oxford Nanopore makes a sequencer called the Minion that is literally the size of a USB thumb drive. Scientists have taken these portable devices into polar ice caps, deep tropical rainforests, and even up to the International Space Station to sequence DNA on site. So, why does all of this matter? What does reading this code actually do for humanity? The implications are genuinely life-changing. First, look at personalized medicine. In oncology, doctors no longer just treat lung cancer or breast cancer broadly. They sequence the specific DNA of the tumor itself to see exactly which mutations are causing it to grow. This allows them to prescribe targeted therapies designed to destroy those specific cancer cells while leaving healthy cells untouched. Second, consider infectious diseases. During global outbreaks, scientists use rapid sequencing to track how viruses mutate in real time, helping us design effective vaccines and pinpoint transmission routes within days, rather than years. And third, look at agriculture. By sequencing the genomes of ancestral wild plants, scientists can identify genes that provide natural resistance to extreme heat, pests, or drought, and breed those traits into modern food crops to safeguard the global food supply. We have journeyed from Frederick Sanger manually reading glowing bands on a gel plate, to massive parallel platforms illuminating billions of data points at once, all the way to pulling a strand of DNA through a microscopic protein hole to read electrical currents. We have officially transitioned from an era where biology was a mystery, to an era where biology is an information science. The code of life is open, and we are finally learning how to read it. If you found this deep dive into DNA sequencing interesting, make sure to smash that like button and subscribe to the channel. Which generation of sequencing technology blew your mind the most? Is it the massive parallel processing of NGS or the electrical current tracking of nanopores? Let me know in the comments below. Thank you so much for watching, and as always, keep questioning, keep learning, and I'll see you in the next video.